right, next presenter is Xin Zeng.
Yeah, Xin Zeng.
Xin Zeng.
Yeah.
All right. His talk is titled, he's from the Oklahoma State University. His talk is Gene Networks Associated with Tillering Traits in Switchgrass.
Good afternoon, everyone. Previous study has shown that there's a positive correlation between the tiller number and the biomass area in switchgrass. In this study, we're interested in identifying the gene networks associated with these tillering traits. Switchgrass shows severe variation as respect to tillering. In this picture, here shows the variation of tiller number between two plants inbreeded from the same single parent. And the Affymetrix chips developed at the Noble Foundation will be used for the transcriptome study here. And we hypothesize we can identify the key regulator factors and the signaling genes that are associated with the tillering traits. The high and low lines of plants from the field was harvested for this study. And the buds and nodes are collected and RNA isolation was done. And the Affy chips hybridization was also done at the Noble Foundation. And there are a total of 8 chips with replicates. And our statistical package was used for the data analysis. As a result, we didn't find a big difference in the number of differently expressed genes between the low and high tillering lines. Certainly in nodes, the number are less than buds. Then we analyzed the transcriptome differences between nodes and buds. We found about 1,500 of them are common. And here, the red color shows the upregulation and green color shows the downregulation. In addition, we did the gene ontology analysis of these genes. We didn't find any big differences between these two lines as respect to gene ontology. However, we do find that in buds, the gene expression and translation was up, while in nodes, the cell death was up. In conclusion, Affymetrix chips with more than 100,000 prop sets was useful for studying the transcriptome. Since nodes and buds belongs to discrete cell types, the gene expression profile, even within the same line, varies a lot. Even though we didn't find any gene ontology associated uniquely with low or high tillering lines, we do find more than a dozen of genes which shows significantly expression pattern between the low and high tillering lines, and the QRT-PCR are on the way to study them. And thank you all.